Good day. My name is Antoinette, and I'm here to talk to you today about phenotypic and molecular characterization of the Marañon genetic group. This is a collaborative work involving staff at CRC in the descriptor team led by Mrs. Frances Vick-Haley, as well as other teams of CRC. And phenotypic characterization, for those who are unaware or unfamiliar with the term, refers to traits that we can see, such as the shape of the pod, the size of the pod, the weight of the pod, or the color, and similar for the beans and other traits such as flowers and leaves. So this collaborative work, the objective is basically to look at the Marañon genetic group and determine whether there are any interesting traits that will make them suitable for breeding. And so just a brief background of the Marañon. You can see here pictured Dr. Francis J. Pound. He was the collector for Cocoa Research Scheme back in 1937 when he did the first collection expedition to collect cocoa accessions in the Loreto region of Peru. For this first collection expedition, Dr. Pound would have taken pods from trees where he observed no evidence of witch's-broom disease along the riverbank of the lower Rio Marañon. So pods were collected by him from between 7 to 20 trees and brought back to be established in Trinidad and Tobago. On his second collection expedition in 1942 to 43, he collected budwood from the trees that he would have collected the pods from in his earlier expedition. These are called mother trees. In total, 277 plants were established at the Marfa farm in Trinidad after quarantine at the Barbados Cocoa Quarantine Station, somewhere post-1943. Currently, most of the carinari, as they are called because of the area where they were collected, are now conserved in the International Cocoa Gene Bank Trinidad, or the ICGT, and the number of PA plants we have are 120. Because the PA are exclusively members of the Marañon genetic group as established by Mukamaya in 2008, we tend to use Marañon and PA interchangeably, so just be aware of that. In addition, as you can see from the map, the PA was sourced from a very wide geographic area, which may explain the vast diversity that we will see as the talk proceeds. So let's look at the morphological traits, which are the phenotypic or physical characteristics or descriptors. From Pound's 1938 report, we noted the general descriptors as follows for pods. The pods were long and generally warty. They had pronounced bottleneck and a conspicuous point. Some pods were almost smooth with 5 shallow furrows, and the range of color for the pods was from pale green to pure green, pale green being false blanco. And Pound distinguished 6 morphotypes for the carinari clones. You can see a screenshot of a paper that was published by Mrs. Vick-Haley in 2008, indicating the 6 morphotypes and a detailed description and how they compare with Pound's categories for pod shape. Next, let's look at some pictures of the types. You can see here there are some distinct differences among the types in terms of the coloring, in terms of the presence or lack thereof of furrows or a constriction or bottleneck, as well as a pointed end or apex. Some pods are smooth and some are not. They are generally warty. And there are some differences physically that you may not be able to see, which is the size of the bean in type 5. So type 5 pods tend to have small beans. And so Dr. Vick-Haley did, Mrs. Vick-Haley did a PCA, a principal component analysis of the carinari clones to show the association among them based on their phenotypic traits. So she would have looked at 11 qualitative and 12 quantitative descriptors of traits. And so this is the result of that data analysis, and the, the principal component analysis, or PCA plot, shows the diversity in two planes, and the clear spread that you can see here indicates that there is a vast array of diversity among the PA accessions or within the Marañon genetic group. The first and second components account for 33% of the total variation seen. And here is an overview of the Marañon, or the PAs, as based on almost the total number of accessions we have in the gene bank. So this is 118, and you can see here the mean statistics for pod length, as well as the standard deviation and the range. And also for the other traits such as pod width, number of beans, bean weight, which is the weight of the seed, or length and width, as well as the pod index and the colors of the pods. There are other traits that we have data for, but it is too detailed to be presented here, and you are welcome to write to us if you would like to have more information on this. In terms of molecular characterization, I have just a snapshot of the genetic diversity within the Marañon. And so here is a diagram that we call a phylogenetic tree. To obtain this diagram, accessions in the International Cocoa Gene Bank were subjected to DNA extraction. So leaf tissue was extracted for DNA, and the DNA is subjected to SNP analysis, or analysis using single nucleotide polymorphism markers. These are genetic type of markers. And so this is the work of the genomic team. From the data generated, we were able to get this plot that shows the diversity of all of the accessions that were looked at. And the yellow area indicates the coronary, or the Marañon genetic group, which group, and they all group together. As you can see, there are no yellow parts anywhere else in the diagram. And as a closer, if we look at it more closely, you can see the groups that are most closely associated, or more closely defined as relatives of the PA, or in terms of the comparison among all the genetic groups, and these are the French Guiana and the Amelonado clones. Let's look at evaluation in terms of yield potential with also some information on disease resistance. So the PA71 is one of the outstanding accessions here because of its good resistance rating for black pod. So this is the pod picture you see here. There are other most promising accessions as listed here in terms of pod index, and PA157 is also notably resistant, or has a good resistance rating for black pod. In terms of yield potential, the next best are these accessions listed here, and here's a picture of PA124, which is among one of the more distinctive accessions. So that we have the asterisk. The pod index range for these accessions would be between 25 to 30.1. In terms of disease resistance for black pod, which is rumen pod diseases, we have for black pod and which is rumen, these 4 accessions all have all show resistance to both these diseases in Trinidad and Tobago. We also have PA46 not shown here, which has resistance to black pod disease in Trinidad and Tobago. And now if we take the descriptive or the phenotypic traits and combine them with the resistance to black pod disease and generate a principal component analysis plot, we can see a very in a way that shows you more graphically which accessions are more standout. So the most favorable ones are circled in red, and that's PA71, PA4, and PA157. These accessions have a pod index of between 24.9 and less than 24.9, and a bean weight of greater than 0.9 grams. The next best would be the accessions that are circled in blue, and they have pod index within the range of 25 to 30.1. And the ones in green are the next best. So this PCA plot is based on 23 phenotypic traits. And so PI in the diagram refers to pod index. And pod index, if you are not aware, refers to the amount of beans required, the amount of pod, required to obtain a mass of 1 kilogram of beans. And so again, you can see a spread of the diversity as in the previous PCA plot, indicating consistency within the results of the analysis. And here is one particular standout accession, and you can see the qualitative description as well as the quantitative trait, which is the numerical measurements that we make on these accessions. And so PA169 has a decent pod index of 29, but it's most significant in that it has tolerance to foster pod disease, as observed by our colleagues and sometimes collaborators in corporate research at the Cartier Institute. Let's look at evaluation in terms of flavor notes. And for this, we would look at gas chromatography, mass spectrometry analysis, as well as sensory assessment. So here is an overlay of chromatograms for this genetic group, the genetic cluster of the Marañon, as analyzed by my colleague, Ms. Naila Ali, soon to be Dr. Naila Ali. So the peaks represent the volatiles, which would have been released for different times along the fermentation progression. So the blue represents day 0, which basically means unfermented. The orange would have been at day 4 and the green at day 6. And you can see there are distinct differences in the volatiles that are produced along as the fermentation progresses. And so after this analysis was done, the next step for Nyla would be to look at the literature and see what aromas are associated with the compounds that were identified via GCMS. And so here you see some of the aromas that are associated with those compounds as dictated by the literature survey that she did. Overall, the flavor volatiles detected in Marañon had predominantly floral and herbal associations based on the literature. And after this was done, next the samples were fermented optimally for 7 days, and then the cocoa liquor created was assessed by a trained sensory panel to score based on the flavor attributes listed in the table above. And so based on that sensory analysis, it was concluded that the liquor had predominantly cocoa flavor with some floral and woody notes. The diagram you see below the table is a spider plot, which is more of a display of the same attributes in the table, but in a manner that you can see more visually. For those who are not familiar with regard to what cocoa liquor actually is, it is the roasted milk that is grounded into a coarse piece. And so let us move on to pulp characteristics. This is work by our colleague, Ms. Rina Kalu, who did chemical analyses of pulp for her master's research. And we had some interesting findings in terms of pulp yield and quality. So Marañon and two groups of refractarios were studied, and it was found that the group with the highest yield was the Marañon genetic group. And within the Marañon group, we had PA107 and PA70 having the highest pulp juice yields. And the cacao pulp yield was determined by hand squeezing of the beans. And if you want more information about how this is done, please feel free to contact us, and we will put you in touch with our cacao pulp experts. In terms of pulp quality, there were significant differences among the PA accessions for pH, as well as vitamin C content of the pulp. So the notable accessions among the Marañon would have been PA169, which you may remember was mentioned earlier with regard to disease resistance. So PA169 has the highest pH among all of the accessions that were tested, and in terms of genetic groups, the Marañon genetic group had the highest pH among the groups. Also, for reducing sugars, we have a PA also being distinguished, the PA51 having the highest level of reducing sugars. But notably, the Marañon group as a whole had the lowest amount of reducing sugars among the groups that were tested. In terms of vitamin C, which is something that people may be interested in for health reasons, we have some distinguishing accessions that are clearly distinct from the others, such as PA51, which is significantly distinct from PA32, 169, and 120, and PA169 is significantly different from PA17 and PA70. So these differences would be useful in determining what accessions or groups may be useful in terms of value-added products involving cacao pulp, such as pulp honey and other products. In terms of the use of the Marañon or the coronary in cocoa breeding, let's look at some reported instances within the literature. First, the international clone trial that was part of the CFC ICCO Biodiversity International Project on cocoa germplasm utilization and conservation, a global approach. This trial involved 4 PA accessions, and it is notable that one of the accessions, PA150, would have been mislabeled in some sites. And so this is something that needs to be considered. In terms of our national breeding program at the Ministry of Agriculture, PA39 and PA46 were used, and these have moderate to high Richardson disease resistance. Also mentioned previously, PA46 also has black spot resistance. PA121 with a part index of 31 in Trinidad and Tobago and Malaysia was also used, and this is something that is also used in Brazil for breeding. We also have PA157 and 159, as well as the distinctive PA169, which has been used in Trinidad and Tobago, and is putatively tolerant to cross-spot disease. Across of PA169 and ICS1 that is resistant to black spot disease is notable. In terms of other national breeding programs, the PA7 has been found to be useful by the Cocoa Research Institute of Ghana, and so they have been using it because of the pot resistance to black spot disease being moderate to high. And so our current objective, which we are working on, is to provide a field guide to the Marañon group or the coronary cacao clones, incorporating all of the information that we have gathered. And this will be an important resource intended for the toolkit of all users of cacao genetic resources. And as I conclude, I would like to share some highlights of what has been found in terms of the Marañon or the coronary. There are 65 promising accessions with pod index less than or equal to 30. There are some distinguishing accessions such as PA71 and 157, having a pod index of 20 and 23 respectively, and also having fair tolerance to black spot disease. And it is notable that neither of these have been used in breeding at this present time. PA169, which you would have heard several times, combines resistance to cross-spot disease with distinctive characteristics that impact final flavor. PA4 is moderately resistant to black spot, as noted in Brazil, and is also reportedly resistant to Richardson disease in Trinidad. So in conclusion, the Marañon genetic group provides a valuable genetic resource for cocoa cultivation and breeding. And now I would like to thank the following. In particular, the support that we get from the government of the Republic of Trinidad and Tobago and the Cocoa Research Association, UK, as well as our director, Professor Kat Umaharan. Additionally, we would like to acknowledge the work of the late Dr. David A. Iwaru, as well as my CRC colleagues, and at the UE St. Augustine campus, as well as those who are stationed at the International Cocoa Gene Bank, Trinidad. I must mention Mrs. Romina Umaharan and also Vinder Singh. And in terms of support for the GCMS analysis, we thank our colleagues at the Hamburg University of Applied Sciences in Hamburg, Germany. For any follow-up, please email francis.bekele@sta.uwi.edu. And thank you very much for your attention. And if there are any questions, I will be happy to answer them now. Thank you.